Hello everyone, I am Elena and I welcome you to our third and final session of the webinar series, Plants as Grandparents, Doctors and Chefs by GeneSprout Initiative. I would like to start by thanking the American Society of Plant Biologists and Plantae for hosting us today. We remind you that today's webinar will be recorded and made available via the GeneSprout YouTube channel and Plantae YouTube channel. So today we are in our third session of the webinar series, and we stand on the shoulders of giants here today. In our first session, we spoke about plant breeding and its history, so plants as grandparents. Last Tuesday, we explored plant health and their medicines. And this session today will all be about plants as chefs, so more broadly on plants for human health. I am Elena Del Pu, and I will be your moderator today. I am a master's student in plant sciences at Wageningen University and member of the board of GeneSprout Netherlands. I come from Italy and I graduated in agronomy at the University of Pisa as honor student at Sant'Anna School of Advanced Studies. So today I'll have the pleasure to guide you through today's three amazing presenters and presentations. Okay, so plants for human health. Because if we want to obtain a more sustainable agriculture, farmers will have to produce more food with less. But how can this be achieved? Three young researchers will try to answer this by giving us a taste of their research on how plants could tackle these challenges. So I first welcome our three speakers today, Maarten, Ramon, and Michiel, young scientists from GeneSprout Initiative. Maarten, in his presentation, will deal with how biotech can tackle global hunger. Ramon will show us how we can make our crops more nutritious from a consumer's and farmer's perspective. Michiel will present what phytoremediation is and how, with this method, we can erase some of the man-made mistakes and hazards to our health. So first, I will introduce to you GeneSprout Initiative. GeneSprout is a collective of young plant researchers based in the Netherlands and in Belgium. Their goal is to increase public engagement and awareness about new plant breeding techniques and their significance in agriculture. GeneSprout provides open access information so that people can shape a truly informed opinion on the future of agriculture. They do this by bringing the fascinating world of plant sciences to the public and by inspiring people to be part of the conversation. As an organization, GeneSprout organizes discussions, workshops, and debates on these topics. One example is the campaign Give CRISPR-Cas a Chance, aimed at informing the broader public on CRISPR-Cas technology and its potential applications in agriculture. GeneSprout is also active on social media, where it continues with its mission in science communication by making science fun. We have placed some direct links to our profiles in the public chat box, the one on the left. We will be glad if you could interact with us via these platforms. And of course, feel free to reach out to us via these channels. So now I'll give the floor to our speakers, but first, let me point out that at any time after and during each of the presentations, you can ask questions via the Q&A box at the bottom right of your Zoom window. You can also post messages of general interest in the public chat left of the Q&A box. In general, we will address all the questions at the end of the three talks, but we'll keep an eye on the chat if something compelling comes up. So let me introduce the first one of our speakers, Maarten. Maarten is a finishing PhD in plant molecular biology at KU Leuven in Belgium. His work deals with biosynthesis of ethylene. He was introduced into the beautiful world of molecular plant biology and genetics during his bachelor and master degree at KU Leuven. As the first PhD student in his lab, he was offered the chance to help with the start of a new research group from the ground up. This offered him many challenges, which granted him plenty of opportunities to learn and improve. Today, his presentation is titled Hunger, Why Do We Eat Plants? And he will frame how biotechnologies can help us tackle global hunger. So welcome, Maarten, the floor is yours.
Hi. Can you see my screen clearly?
Yes, we
Okay,
say
thank you.
great.
Yeah, thank you, Elena, for the kind introduction. So indeed, normally I work on ethylene in plants, but for today I came a bit out of my comfort zone and I read up on some important topics that are sadly enough not getting the worldwide attention they deserve. And therefore I titled my presentation, Hunger, Why Do We Eat Plants? Because in the end, our food web starts with plants. Um, I want to give a quick reminder of how plants themselves get their food. So, and how actually our food web starts. Plants, unlike animals, do not obtain organic material for their nutrition, although these form the bulk of their tissue. By trapping solar energy in photosynthetic systems, they are able to synthesize nutrients from carbon dioxide and water. However, plants do require some inorganic salts, which they absorb through the soil surrounding their roots. These include macronutrients like nitrogen or potassium, but also some micronutrients like chlorine or iron. So, like most of you will know, humans transitioned from hunter-gatherers to a more agrarian civilization. And the earliest evidence of this can be traced back to some 13,000 years ago in the Middle East. And this transition allowed for the dawn of human civilization as we know it. You have agrarization, urbanization, development of early cities, but with this, humans also started domesticating plants and unknowingly selecting and changing different plants to fit human needs. And for example, they chose fruits that were not lying on the ground, but that were still on the plant, selecting therefore for fruits that no longer abscised. Or they made sure that there was a loss of fertilization requirement, therefore that you had a flowering more easily, more readily. In general, you could say that there was a reduced seed dormancy too. You could actually say that big changes in humans led to big changes in plants. Now, plants undoubtedly play an important role in our diet. Not only are they the healthy part of our meals, providing nutrients, vitamins, minerals, fiber, and water, but they are also the basis of our entire food web. However, by 2050, we need to feed between 9 billion and 11 billion people, according to some estimates. And while the UN has vowed to end all hunger by 2030, we have seen that we've seen a recent increase in malnourishment and undernourishment since 2015. And this can, well, this does not include the problems that we might face due to the huge impact of climate change. Um, by 2050, we would technically need 4 Earths to support our food demand, while we might only have access to, well, barely one fully functioning Earth. This does obviously not bode well. We therefore need to ask ourselves, is the human food supply in danger? Well, many research groups are trying to predict the effect of what global warming will have on crop yield. However, this is obviously not an easy objective. There are an incredible number of different parameters that must be considered, and I will try to simplify it a little bit here to 2 main points. And first off, there will be a direct temperature effect on the crops themselves, which, like many separate research groups have shown, will have a major direct effect on crop yield. Well, basically, most plants do not like a sudden change in temperature. And we must remember that we have to consider the evolutionary timeline, and this temperature change from the last few decades is sudden. Second, the increase in temperature will cause an increased demand in atmospheric water, leading to a decreased soil water content, and in turn resulting in a decreased availability of water for plants. And even when we only consider these 2 factors, we can presume that in the future, with moderate to severe climate change, the growing periods will be a lot shorter. Simultaneously, the prevalence of pests and diseases will rise, plaguing plants that might already be suffering from abiotic stresses. And again, These problems have already been on the rise since 2015, which you can see on this graph, showing the prevalence of undernourishment in Western Africa. And many of the problems with the food supply can be traced back to human conflicts, however, not all. As you can see in this graph showing the prevalence of undernourishment and the estimate number of undernourished people in sub-Saharan Africa, droughts also heavily contribute to the problem with undernourishment. Um, but well, food insecurity is more than just hunger. The majority in the world have adequate access to food in both quality and quantity. But on the other side of this spectrum, you have people who experience severe food insecurity, meaning they might sometimes go days without any access to food at all. And then there's a middle group that has also been growing. These consist of people who experience difficulties to obtain food, but in general terms still have access to food. But they do have to compromise on the quality of the food they consume on a daily basis. I want to go in a little bit deeper on this undernourishment and malnourishment. Um, these are the numbers from 2016, and back then almost 800 million people were suffering from hunger. The latest numbers from last year haven't changed that drastically. By estimate, there were still 750 million people suffering from hunger. So that was the red in this graph. And but at the same time, there is now 1.2 billion people suffering from moderate food insecurity. And these people have to compromise on their food quality. In total, there are 2 billion people who suffer from malnourishment of some form, translating into micronutrient deficiencies. And these people suffer from what is called hidden hunger. Hidden hunger occurs when the quality of food people eat does not meet their nutrient requirements. So the food is deficient in micronutrients such as the vitamins and minerals that they need for their growth and development. Especially women and children in families with low income often don't get enough vitamin A, iodine, and iron, and sometimes other essential nutrients. This limits their growth and development, but also their health and working capacity. And this is partially due to a lack of nutritional density in modern crops. But on the other hand, there's also a lack of agricultural and dietary diversity. The solution for these problems starts with better education. Education is actually the key here. Introducing farmer schools and revising educational programs and at the same time having researchers collaborate with local farmers to ensure better farming practices. A way for biotech to help in this problem is to develop and promote new, more nutritious varieties of staple food crops, with higher amounts of, for example, vitamin A, iron, or zinc. Three of the micronutrients that were identified by the WHO as most lacking in diets globally. This is what we call biofortification of crops. And this is, however, not the solution to world hunger, but it complements the solution. Harvest Plus is, for example, an organization that helps to lead the effort in this regard. They have already developed so-called better seeds, better plants containing several different micronutrients, for example, iron, beans, and pearl millet, vitamin A, maize, cassava, and sweet potato, and zinc, wheat, maize, and rice. How do they do this? Well, they start this process by screening wilder varieties of commercial crops for a desirable trait. And then they try to pre-breed these interesting ecotypes with more commercial variants to transfer these desirable traits. Because a lot of the wilder ecotypes are, um, well, still have many undesirable traits. For example, you might have seen pictures of bananas containing many, many seeds, which render them unedible. Since many of the wild varieties um, have not gone through a, well, proper domestication process. After further optimization and breeding, they will end up with a potential interesting cultivar, which they then test in experimental stations and together with local farmers on the fields. They need to evaluate how this new variety tolerates pests, stresses, and diseases. And whether they yields they get in experimental conditions is translated to the field. After this, the new crop variety is evaluated and released to the growers. Well, this entire process takes about 10 years. And this is why more recently biotech has started to get more invested by using transgenic approaches to quicken the whole first part of the process. And they then directly take a desirable trait and transfer the responsible genes to the host crop. Other than the time gain, this has also other advantages. For example, the possibility for the so-called multi-biofortification of food crops. In this approach, genetic engineering strategies deliver multiple vitamins as well as minerals, but also other interesting agronomic traits, for example, leading to higher yield. Um, in the, well, are included in that biofortification design. In ideal cases, these designs can be universally applicable with, well, with relatively little difficulties. Genome editing tools can be used to guide the genomic insertion to a well-characterized and safe harbor region in the crop's genome. And in doing so, resilient, nutritious crops of high agronomic value can be obtained, hereby helping in the effort to reduce malnourishment and undernourishment. With this, I conclude my short talk, but don't worry, in a minute, Ramon will tell you more about crop improvement. And well, with this, I would like to return the words to Elena.
Thank you very much, Maarten, for your presentation. You know, this is great because we often think that biotechnologies deal with very complex and far-reaching stuff, but here you showed how important and basic needs can be fulfilled via biotechnological tools, and also what is it at stake if we don't use them. And of course, as you yourself said, we don't have a solution, but we can indeed show a possible set of complementing solutions. So thank you very much, Maarten, for your presentation. I also remind the audience that whenever you have a question, you can pose it in the Q&A button on the bottom right of your Zoom screen. Please be patient. The questions will be answered by our three presenters in the Q&A session at the end of the three talks. So now I want to introduce to you our second speaker. Ramon is a fourth-year PhD researcher and teaching assistant in bioengineering sciences at the Free University of Brussels, VUB. From an early age, Ramon was fascinated by the natural world around him. His curiosity and excitement to know how everything works made him pursue his bachelor in bioengineering at the VUB in Brussels. During this time, he discovered the fascinating world of plants, which led him to his master's in bioengineering, specializing in agrobiotechnologies at the VUB and UGent. His research focuses on the use of CRISPR to enhance the nutritional value of the common bee. Ramon is also very much committed to science communication, and this is why he joined other PhD colleagues in starting the Belgian branch of GeneSprout initiative. Today, Ramon's presentation is titled Let's Improve Our Crops, and he will give us a biotech perspective on nutritional enhancement. So welcome, Ramon. I will leave you the floor.
Thank you very much for those kind words. I will share my screen. All right. Okay. So welcome everyone. Today's talk will be about let's improve our crops. So yeah, let's go for it. So first of all, how does a superfood look like? And I gave it a try, and I came across this with a cape and a Superman bandage. But on a more serious note, we have to take into consideration different perspectives. And one of them is from the consumer side and the others from the farmer side. And so from the consumer side, what kind of traits do we want in our superfood? Well, of course, we want to have our food very tasteful. We want it healthy. We also are very picky, so we want to have it look good. Shelf life is also important because we want to store our food long enough. We don't like mold on it. And nowadays, it's more and more important that it's coming from a sustainable source. From a farmer's perspective, it's always very important that there is a high yield. Farmers also want to use less fertilizers, and also water is getting more and more scarce, so we want to lower the input. It's always best to have a crop that's pest resistant, so the farmer doesn't have to spray. And we also want to have our crops climate resistant, and especially also because Maarten talked about it a bit, about drought getting more and more serious, and also flooding in Asia, for example. So we want our crops to be adapted to that. And that's not so easy to put all these traits into one crop. Why not? Because it's a bit of a trade-off. And here you can see like the strawberry, in which Priska talked in our second webinar. And on the left side, you can see the defense system of the strawberry. And on the right side, the growth. And so basically, it's a bit of a balance. If you want to have a good defense system, the plant needs to put a lot of energy into that. And then there is less energy available into the plant growth. And on the other hand, if we, for example, want to have strawberries which contain a lot of sugar, they are more prone to molds, so the shelf life will decrease. Also, I want to note that this is not rocket science. I mean, in the sense that we are actually already doing this for 10,000 years. Maarten already said a bit about it, and it's only about like the recent 100 years that we are doing this in a more precise and effective way. But we are already doing it for a long period of time. So if you want to improve our nutritional quality of our crop, how can we do this? And one way is by increasing the beneficial compounds. So if you have a look at, like, for example, beans, here you see on the top the micronutrients. So they contain quite a lot of vitamins, especially vitamin B, and also quite a lot of minerals like copper, manganese, and iron. So that's already something good. And if you look at the other micronutrients like proteins, carbohydrates, and dietary fiber, we can see that they also contain quite a lot of protein and they have a high dietary fiber level. And so basically we can enhance the amounts of these nutrients even more. And for example, in our lab, we work with the common bean, but also soybean. And if you can see here on the left, you can see kidney beans, which are a kind of common bean, and they contain quite a lot of proteins. So they're a good source from that. And if you zoom in to the proteins, proteins are built up by amino acids. And 8 amino acids are essential for us humans. So that means that we cannot produce them ourselves, but we have to take them up from our food. And you have like a differentiation. There are some that are produced in grain crops, like for example in corn, and others that are produced in leguminous crops, like for example beans. And you can here already see that there are some only produced in grain crops and others only produced in leguminous crops. And that also states that our diet needs to be varied, so we need to eat both. And for example, in our lab, we want to increase the lysine concentration of it. And how are we going to do this more in detail? On the left side, we see the pathway, the metabolic pathway to produce lysine. This is a very global view of the pathway. It's more complicated than this, but it gives a general idea. And in this pathway, there's a key enzyme called DHDPS. And that DHDPS is actually presented here in gray. And an enzyme is actually a kind of machine that transforms one substrate into something else. So in this case, it transforms aspartate for semialdehyde, which is like depicted in green. And it transforms it into 2 small molecules, which are then transformed by other molecules to obtain lysine in the end. And so this pathway is in the plant is regulated by a negative feedback. What does that mean? It actually means that the lysine produced can bind to an allosteric site on the enzyme. And by doing so, it alters the form and the substrate cannot bind to it anymore. So actually, it inactivates the enzyme. So by doing so, if you produce more lysine, lysine can bind and the production is halted. So how can we increase this? So if you look here, we can actually alter this. What we did in our lab is we altered the site where the lysine can bind. So this is depicted by the triangle. And so what happens now, lysine cannot bind anymore. So the feedback is stopped. And because of that, the enzyme can keep on producing the lysine. So the lysine concentration enhances. So we get crops with higher lysine concentration. Another way to improve our nutritional quality of our crops is by reducing or removing the anti-nutritional factors. So there are certain things present in the crops as well, which are considered anti-nutritional. So some of them are presented here. And if you have seen our second webinar, maybe you could see that like these three here, lectins, saponins, and phenolic compounds, that they played a role in the plant defense system. And that's actually correct, but they are actually also sometimes considered anti-nutritional because lectins and saponins can sometimes be toxic for us. And phenolic compound, for example, an tannin, can decrease the digestibility by binding to proteins. So another one is phytic acid present in the seeds. And what does that do? It actually can bind to dietary minerals like calcium, iron, and zinc. And by binding to them, they are not longer available for us, so we cannot take them up anymore. So our uptake of these dietary minerals are lower. And then in the end, like there are also enzyme inhibitors, which reduce the digestibility in general. So here I want to emphasize as well that it's also, again, a bit of a balance, like between, on the one hand, the plant defense system, and on the other hand, the nutritional quality. So what I'm working on myself is, for example, this is a seed of the common bean, and there are some oligosaccharides present, raffinose and stachyose. And these oligosaccharides have an alpha 1.6 haloxylic bond, which are depicted here in red. And we humans and monogastric animals don't contain the enzymes to break these down. So what happens? They go through our digestion system and they end up in our large intestine. And there are microbes present which do contain these enzymes needed to break these down, but they produce gas. And so we all know that if we eat beans, that we get flatulence and sometimes even stomach discomfort. And to reduce that, we want to lower the amounts of raffinose and stachyose in the beans. So on the left side, we see here the metabolic pathway in which raffinose and stachyose are formed. And so basically, the enzyme which does this is raffinose synthase, here in gray. And the substrates are galactinol and sucrose. And so they can bind to the enzyme, and the enzyme will then link them, and raffinose will be formed. So in order to produce less raffinose and stachyose, the easiest way is to just knock out raffinose synthase. So basically so that like the plants don't produce this enzyme anymore, and so these compounds are not formed anymore. And how do we do that? Then we actually refer back to like the first webinar where Nick talked a bit about this already. We use new plant breeding technique called CRISPR, which is actually a short kind of search and replace tool. And on the right side here, you see the genetic code, like our DNA containing nucleotides. And these are actually the building blocks of enzymes. And so if we just make a small change, like for example, a deletion of a C, this can also already like make the enzyme not active anymore. So that's what we are trying to do in our lab. And what happens then? If you change that, the raffinose synthase is not anymore produced. And like this enzyme is just not present anymore. So we will not form these raffinose and stachyose. So we get a lower amount of them. So these are some examples of how we do this more in practice in our lab. There are, of course, many other things that we can do, but it's to give you already a bit of a taste of what we're doing in our lab. So thank you very much for your attention, and the floor is yours, Elena.
Okay, thank you very much, Ramon, for your presentation. What I think is great is that we had two very complementary presentations on the topic of how biotechnologies can improve the nutritional value of crops. And while before with Maarten, we spoke about a hugely important global goal of combating hunger, not only for sustainability, but also in terms of equity of countries and human rights. Here we went through how the same instruments can tackle the daily goals for farmers, for small businesses, and for consumers as well, of course, which is something that touches our daily lives, I would say. So thank you very much again, Ramon, for your presentation. I again remind the audience that whenever you have a question, you can pose it using the Q&A button, and be patient, the questions will be answered at the end. So let me introduce now our third and last speaker. Michiel is finishing his PhD in plant physiology at the Center for Environmental Sciences of Hasselt University in Belgium. His research involves studying the effect of metal cadmium on the leaf growth of rice plants. During his master's degree in biology at the KU Leuven, he developed a keen interest in biotechnology and plant sciences. He's a strong believer that the usage of biotechnologies can help in safeguarding a sustainable future. In 2019, he joined the GeneSprout initiative as the 5th Belgian board member. So today, Michiel will present to us phytoremediation, plants cleaning up our soils, where he will show us what plants can do to remedy human mistakes and environmental as well as health hazards. So welcome, Michiel. I will leave you the floor.
All right, let me just share my screen. All right, let me get my pointer. All right, so Elena, thank you for this nice introduction. Maarten and Ramon already talked about how we can improve the nutritional value of our crops. Well, I will be talking about something a bit different about phytoremediation and how plants can help us clean up soils. So today I will be mainly be talking about metals in the soil. So how does these metals come in the soil? Well, there are multiple anthropogenic sources. First of all, you get atmospheric deposition coming from industrial activities or the combustion of fossil fuels. You also got irrigation of the field with water coming from mining facilities or the usage of sewage sludge. And lastly, you also got the usage of fertilizers, where mainly phosphate fertilizers are known to contain a lot of unwanted metals. So how does it affect human health? Well, this is a very extreme example of how things can go wrong. This person here is suffering from the Itai disease, which is caused by severe cadmium poisoning. And this happened somewhere in the early 20th century in Japan, where mining facilities were dumping their leftovers into the Yenzu River. And then further downstream, the farmers were using this water to irrigate their rice fields. And so the cadmium accumulated into the rice grain, and this got eaten by the locals. So how can we clean these soils? Well, in Japan, the main method was soil dressing for nearly 8,000 of hectares. And this means that they replace the soil, so the contaminated soil, with uncontaminated soil, or just burying it underneath. And this is actually a very efficient method when the new soil layer is sufficiently thick, but it also has some problems. And one of these problems are that it's becoming harder and harder to find unpolluted and also fertile soils. Another solution would be soil flushing, and this involves pumping a solution into the contaminated soil. And for the extraction of metals, this mainly is a diluted acidic solution. And then afterwards, the contaminated groundwater is again pumped up and the metals can be extracted. Now, this is also very effective, but if you just look at the setup, you can immediately see that it's very costly, labor intensive, and time consuming. And to be honest, it also causes quite a disturbance of the microflora and to the soil itself. So a more ecological approach would be phytoremediation. And there are also different strategies that you can deploy. For example, you got phytostabilization, and here heavy metal accumulating plants will immobilize the metals into the soil so that they are prevented from leaching into the groundwater. You also got phytodegradation and phytostimulation, and these are actually technologies that are more for organic compounds because metals cannot be degraded. You also got phytovolatilization, and this is also more for organic compounds, but there are some metals like arsenic and mercury for which this could be used. And then finally, you have phytoextraction, and this is a very attractive solution, I think, because you will permanently remove the metals from the soil, and our plants functioning as chefs will here become plants functioning as ground workers. So now, what is an ideal plant usable for phytoextraction? Well, you can immediately see that this will not be an easy task to find such plants. Of course, a high metal extraction capacity is needed, but also a high metal tolerance, because otherwise your plants will start to suffer and die eventually. It also needs to be fast growing and have a lot of biomass, because small plants that accumulate lots of metals will still not get enough metals from the soil. Excessive root system, of course, to penetrate the soil and to cover enough ground. Your plants also needs to be easy cultivatable, and the harvest should not be too costly. And then finally, repulsive to herbivores, because when your plants get eaten by animals, then yeah, that's another way of how metals can enter the food chain. But this first characteristic is probably the most important one. So these metal hyperaccumulating plants are plants that have a natural tendency to accumulate large amounts of metals. And here are a few examples. Sedum alfredi is a herb from Asia, and it actually has the potential to accumulate more metals at once. And it's also used a lot in research to investigate the molecular mechanisms of metal uptake. Sunflower, it is a well-known plant, but it can also accumulate up to 5 grams of lead per kilogram of dry weight. So that's a lot. And then lastly, Nicotiana tabacum is a hyperaccumulator of cadmium in the leaves. So each cigarette can contain up to 1 microgram of cadmium. So that's probably one reason why you should not smoke this plant. So how do these plants hyperaccumulate metals, well, I will not go into the molecular details, but there are three main characteristics, and one of them is, of course, an efficient uptake through the roots. Then they get translocated to the shoot, mainly through the xylem, and then in the shoot they can be bound, for example, to cell walls or stored into vacuoles. And then, yeah, after the shoot is harvested, it can be used, for example, for the production of biofuels or for burning for energy and heat. And then from the remaining ashes, the metals can be extracted, and this process is called phytomining. Okay, so the discovery that CRISPR-Cas also has the potential to transform plants is also very interesting for phytoremediation. And you can go both ways with this technique. You can, one way is by stimulating the growth of known metal accumulators, such as Sedum alfredi. But this is very difficult because growth is is dependent on many factors such as photosynthesis, nutrient uptake, phytohormones. So another way is to go for already high biomass producing plants and then target individual genes which have a role in metal uptake, but also metal tolerance. And yeah, if I think about fast-growing plants, I immediately think about poplar trees, which also have a huge economic value. And in this paper, they use the CRISPR-Cas system to target a gene, the PDS gene, which is responsible for, it's actually essential for biosynthesis of chlorophyll, and that's the green pigment of plants. So if the transformation succeeded and you get knockout of this gene, you get albino plants, and you can see them here. So what I want to show you is that it's already, these researchers already succeeded to use this system to transform poplar. So a next step would be to target, of course, genes related to metal transport, and then you could get huge trees, which also could have lots of metals in their shoot. Okay, now coming back to the cadmium in the rice problem. Well, I think this is something that can and should be avoided in the future. Let me explain. Well, here you see an illustration of a transverse section of a rice root. And cadmium uptake, well, an important transporter is the NRAM5 transporter, which normally transports manganese, but it also has a high affinity for cadmium. Now, researchers targeted this gene with CRISPR-Cas to create knockout lines of this transporter, so it is not functional anymore. And if you look at this graph, you can see that the cadmium concentrations in the rice grains are much lower in these CRISPR lines when compared to the normal plants, which are the blue bars. So I think that this example clearly illustrates the potential of genetic modification and how it can improve human health. And if you also think a bit more about this example, for example, an overexpression of this gene might potentially have the opposite effect. So you get more cadmium in your shoot and in the grain, and then you might have plants that are much better in phytoremediation. And with this, I would like to end my talk, and I would like to thank ASPP and Plante for hosting and for giving the opportunity. Okay.
Okay, thank you very much, Michiel. For everybody, I also posted the links, both of the social of GeneSprout Initiative and of the LinkedIn accounts of our 3 panelists. But first, thank you very much, Michiel, for your presentation. The topic of the webinar here today is plants for human health, but what does human health mean? How do we preserve human health if we don't preserve our environment's health? So thank you very much for having given us a taste of your research in this area. And thank you to all of you as our 3 speakers. We will now go into the Q&A session, so to the debate. Please, if you have further questions, don't hesitate to post them using the Q&A box. I would like to start with an ice-breaking question, actually, to Ramon. So I saw in your slide you had mentioned the saponins, and I know that saponins are, for example, a problem in the plant breeding of quinoa. So I'm super interested because for what I know, quinoa thrives in very harsh environments, in the highlands of South America at very high altitudes. And so actually quinoa in the wild needs saponins to thrive, right? And as a deterrent for animals, or, just as a protective compound for their seeds. So how do you think, for the farmers and for local farmers on the one hand, and, also for the consumers who don't want to have these anti-nutritional compounds, do you think that a balance might be designed, among the plant needs and the needs, or I don't know, the ease in consuming for the final consumers? So yeah.
That's a whole question. No, it's actually a really interesting question, and it also shows that it's not so easy as what I said already in the slides, as in like it's really a balance between, indeed, on the one side what the plant wants is like a plant defense, and on the other side what we as consumers want. And it really varies. Like, for example, small amounts of these defense molecules can also have a beneficial effect on our health. So it's not to completely remove them that it would be all good. Like, can be that a small amount of them, there is already like shown that like some of these compounds can have like even anti-carcinogenic effects. And for example, also for the raffinose and stachys, like in lower amounts, they can be even good for like the gut microbia as well, because it's an energy source for them as well. So it's really always, you have to take into consideration the whole field, like indeed. And with quinoa, I'm not an expert in quinoa myself, but I can see that like we have to check out how much of these saponins should be present in the plant, that they can still have a beneficial effect for the defense, and what is the threshold of that. And like if we could lower it to that point, it could be even also beneficial for our health as well. But that's something that science needs to figure out.
Yeah, of course. So it's not only a work of removing things from the plant, but also of managing and the balance of the field. Okay, this is great. We have a question for Michiel. So on phytoremediation, it is not clear to me the goal of phytoremediation is to fix the symptoms of agriculture, or if we could also use them to target their cause. Can we use phytoremediation in tandem with more sustainable agricultural practices? So still for I imagine it's still for the pollution in our soils caused by agriculture.
Well, I think it's quite a straightforward application in itself, because and also with genetic modification, it might be actually a bridge to genetically modified crops, because yeah, you can clearly see the benefits that these plants are doing, and yeah, people don't have to eat them, and that's one thing that that people are quite hesitant to do. And yeah, so I think they can really form a bridge to yeah, getting also genetically modified crops a bit more publicly accepted. And what was the second question you had? Or
if they can be matched with more in tandem with more sustainable agricultural practices, so you know, not only solving the you know, when the soils are polluted, so not only as you know, as a solution at the end of the chains, but also to prevent maybe what leaks up from our soils.
well, that's quite a difficult question, because when you have polluted soils, you also have to be careful. And well, but for example, if you can create rice varieties that accumulate lots of cadmium into their shoot, but they will not remobilize them to the grain, well, then you could have like producing rice that still can be eaten, but the shoots you could dispose, and then you would in time also remove the metals from the soil, for example. So that's something that is definitely possible, I think, if you target the right genes, but it's something that still needs to be a lot investigated, because it's not a very easy task to do.
Yeah, so what you're saying, because I remember you work on the cadmium on rice, right? So what you're saying is that we can even like transport the you know, the molecule of the pollution in soil into a tissue that is not the tissue that goes to the consumer. So is it possible 100% to have I don't know, like cadmium going into, I don't know, imagine the leaves and the seeds to be free of anything that is
Yeah, sure. So normal cadmium uptake goes to the roots and then to the leaves, and then during the reproductive phase, the cadmium gets remobilized from the leaves to the grain, and also a bit from the root. But yeah, in this remobilization process, there are genes that you could target to not do that anymore. But then you could have safe rice grains, and your cadmium would still be in the leaves. So when you harvest them, then you could separate the shoots from the grains, and then you have two how do you say it, flies in one yeah, thing to do.
Yeah, wonderful. Wonderful. Thank you, Michiel. Then we have one question from Anik. I hope I'm pronouncing it well. So she says, thanks to all the speakers, super interesting talks. I was wondering about your thoughts on the implementation of such techniques into the real life. I assume people in the general are still hesitant towards the introduction of GMOs into nature, or has it changed in the past years? I think it's a general question to all of our presenters. So I don't know, Maarten maybe wants to have a say. I don't know, we can start from Maarten just because it was the first one.
I think if I have to consider the topic I presented today, it might be different, because in that case you have a necessity. There you it's either eat GMO or go hungry. Well, that's putting it too directly, but I mean, there using GMOs could basically be the difference between a good growth season or going or eating, or having a failed crop and going hungry. So I think that would be my answer here.
I think like humans in general need like some kind of stimulus to really like appreciate something like, for example, a GMO. And I think especially with climate change now, if we get like crop losses and we get like newspapers stating like, again, so much of our crops has been lost and blah, 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 then people realize like, oh shit, we actually should like do something about this, and then completely think about it differently. So I think in the future, people will for sure be more pro, I would say.
Michiel, do you want to add something? Wait, you're muted. You're muted.
Sorry. Well, I agree with Ramon actually, because now most of the time it is not needed to get GMOs, especially not for developed countries. So I think when the need is there, then also the questions will arise: should we start producing them? So yeah, I think people will become...
Hopefully it's not too late.
Sure. Great. Then I have another question, more for Maarten and Ramon. So the first huge topic of nutritional enhancement. It is, how long does it take until CRISPR plants would be on the market? So, maybe more on the market from the Ramon part of the presentation. And I don't know, Maarten, you were talking about global goals. So I imagine, I don't know, goals like the golden rice. For example, how do you think such projects can be put in place, and a real plan can be put up there really to solve global hunger in third world countries?
Golden rice has actually already been produced, for example, in Bangladesh, and there it's provided by the government for free to the farmers. So farmers itself can get those seeds, and the good thing about this is that they have to use way less amounts of pesticides. Oh no, that's actually a BT, but like golden rice is also produced there. But they, for example, also had I think eggplants which are resistant to BT. Well, with BT resistant to certain pests. So, but they are both given for free.
Yeah, but in terms of time, like you work in the lab, what do you imagine would be like the time frame from the lab to the people in this case? But it's more the question.
Yeah, that's still quite long. I mean, because in the lab, it's not as easy as we all think it is. So that takes, I would say, still a few years. And then testing in the field needs to be done because you need to have like a stable variety. So it will be much faster than more conventional methods, that's for sure. But that's why I stated it, like, hopefully it's not too late, because it takes time. And like, if I would put a time limit on it, it would take around 5 years or something. But I'm not sure myself.
Okay, thank you very much. I think today our webinar session is coming to an end. I guess you see my slides back. So thank you for staying with us today and participating so eagerly with the questions. If there are any remaining questions or comments, you can contact GeneSprout via these channels here that you see in the slide, and that I also posted in the public chat box, so that you can directly click in there. Please do not hesitate to contact us, and we will be glad if you could support us by following us via the socials. Thank you for having me here today. Thank you to our 3 amazing speakers, and to GeneSprout for having organized this occasion. Also, my biggest thanks go to Plantae and the American Society of Plant Biologists for hosting this webinar series. And of course, enjoy the upcoming days of rest and holidays.